So thanks for the introduction. Yeah, I'm going to talk a little bit about some different AI and agentic systems for biological research and chemistry research. Um, so just kind of an overview of what I'm going to talk about today. So I'm going to kind of introduce some challenges related to AI research for biology and chemistry in particular. And then I'm going to walk through several different example AI systems. Um, One called Talk to Your Microbe, which is something that is still in development here at EMSL, which is trying to extract biochemical information for microbial systems. We're going to talk a little bit about ChemCrow, which is a tool used for chemistry, an AI tool for chemistry. I'm going to talk about uh, Reactome, which is an AI agent for biochemical reaction pathways. And then I'll talk a little bit about kind of a more general framework that some folks at PNNL are developing for scientific discovery using agentic systems. And then I'll kind of wrap things up with a little discussion. So before I get into the technical talk, I wanted to just introduce myself a little bit more. So I'm a computational scientist, and I kind of focus on developing AI and machine learning methods in the bioengineering domain. And I did a physics undergrad and then biomedical engineering PhD. And I have a couple different research interests across AI and machine learning, which I've listed here. And today's talk is really all about using AI to help with scientific discovery. Um, and I wanted to touch a little bit on how I got into agentic, specifically AI systems. So when ChatGPT 3.5 came out, I was pretty impressed and ended up using it a lot. And then the ability to start making these custom ChatGPTs came out, and I kind of started playing with that, and then eventually kind of worked towards building agentic systems using LangChain and tools like that. And I just bring this up because, there's a lot of different paths I think you can take to get into this type of research, and it's, an emerging area. And so I think the more people who work in it, the better. So, um, biology sort of has a problem with data. And the problem is that now we're able to generate enormous amounts of data each year. So if we just take a look at genomics, so where we get the sequence information of an organism, it's estimated that we'll generate between 2 to 40 exabytes. So an exabyte is a million terabytes per year just on genomic information. This is, up to maybe 40 times larger than YouTube. It's much larger than all the astronomy data that's generated, much larger than all the data generated from Twitter in a year. So when you have this much data, it's hard to be able to, you know, analyze and reason about this complex data. And so particularly finding and retrieving, specific information you want be, it becomes kind of a challenge. And there's a lot of, challenges with biological data, in particular, different qualities of the data. It can go through different kinds of processing steps which you want to track. Trying to find relevant papers or evidence to support some claims can be challenging. There's a whole suite of different types of tools that people use to analyze biological data. And so due to the large data set sizes and kind of complexity of this data, actually extracting information you want and analyzing that for scientific discovery is a pretty big challenge. And so kind of the, motivation, I think, behind these AI systems and agentic systems that I'm going to talk about is how can we use these to help address this challenge of getting biological data that we want for specific scientific questions. If we kind of zoom in a little bit on microbial research, people have spent many years kind of curating data and developing biological databases that organize knowledge in a systematic way. And so this helps with the challenge of, extracting useful information for scientific discovery, um, but it doesn't completely solve the problem. So some example databases are UniProt, which focuses mainly on protein sequences and function. KEGG is another database which focuses mainly on just biological function. And Biopsych or EcoPsych, which is kind of an encyclopedia type of reference and database for biological systems, or E. coli for EcoPsych in particular. And if I kind of zoom in a little bit further on just looking at Biopsych compared to the other databases, it contains a significant larger number of genomes, pathways, reactions, and citations for its curated database compared to a competitor, KEGG. These databases often have tools for kind of downstream analysis as well as visualization. So we have these nice databases to organize information. And one of the questions is kind of how well does a large language model actually know about biochemistry, or in this case, microbial biochemistry? And the answer is not so well, kind of out of the box. So a 2024 paper looked at this and looked at ChatGPT and Gemini and asked it a set of kind of biochemistry questions related to microbial organisms. And I think there was 8 questions, and they kind of had a dialogue back and forth, and were graded on a score of 0 to 3 per question. And you can see kind of at the bottom highlighted that the scores for GPT was about 21%, and for Gemini or Bard at the time, it was about 54%. So not very good, kind of naively out of the box using LLMs to answer biochemistry questions. Some of the brain reasons is that the large language models will hallucinate, so they can make up fake sources if you ask it to give citations, and they can also just make up scientific facts that aren't true. Now, I will say that since this paper came out, newer models and kind of modes of agentic systems have improved. There's anything that's able to do a search on the web now, or kind of a research mode, will do much better. However, these models are still pretty expensive, and there's usually limitations to how much you can use them. So kind of with this in mind, I'm going to move into some example systems of agentic systems. And first, I'm going to talk about something that we're working on at EMSL called Talk to Your Microbe. So the idea the idea of Talk to Your Microbe is to incorporate the knowledge we have from the E. coli database, EcoPsych, so it can be used by a large language model. And this would work. So how this would work would be users would ask a question about E. coli, and the large language model would respond in a natural language fashion. And the kind of benefits of this would be to reduce the barriers to accessing scientific knowledge. It would help extend large language models' capabilities for this kind of niche area of research, and help automate kind of knowledge retrieval tasks. Now, I'm going to say that this is still in development at EMSL. It's currently not public, so just keep that in mind. So the task we have for Talk to Your Microbe is to take a natural language question and convert that into a database query, which can then query the EcoPsych database, which I mentioned, and then retrieve the information from the database, and then output that as a natural language response. So this is a knowledge retrieval task, and there's actually several ways that you can approach this in a large language model system. So I'm going to present two ways that we're thinking about kind of doing this. And I also want to touch on and some other concepts, such as generating benchmark data and kind of thinking about how this can be used within larger AI models and workflows. So the first approach to access data from this database, EcoPsych database, is to use retrieval augmented generation, or RAG for short. RAG has already been mentioned before, but essentially it gives large language models access to additional data, which improves the reliability and accuracy of the outputs of the large language models. So the workflow is to extract data from EcoPsych. EcoPsych is a database which has its own kind of query language that we can use to pull down the information of E. coli in this instance. So we could, say, for example, pull down all the reactions, all the pathways, all the genes, etc., and make a big document with those. Then we can go through and chunk that data into smaller sizes, and then embed it in a vector embedded database. So once that database is created from the information, then a user will ask a question, which will run through the embedding and query the vector database. And there's different ways to do that, but you can essentially see how similar the text that they input is to the text in the vector database store. It'll return that result in a natural language response. And there's several tools you can use, but the main ones we're using are ChromaDB for the vector database, LangChain, and then OpenAI as the kind of backend model, and Chainlit or Streamlit as the kind of front end and UI. Another approach I wanted to touch on because it's an interesting kind of new emerging approach with these models is called structured generation. So the idea between structured generation is to force the large language model to generate an output based on a schema or set of rules. So you can think in a simpler example, you want the large language model output to be a JSON file. So it always has to output a JSON. Or in a more complex or advanced approach, you can even give it a grammar or like a language that it has to adhere to. And so we'll output responses that adhere to a definition of a language. So what this allows a large language model to do is now kind of integrate with a larger workflow potentially that requires kind of a specific format for the output. So for something like EcoPsych, this database has a particular query language called Biochemical, and there is a package called Lark, which can take that query language and convert it into the type of grammar needed for the structured generation. And there's another tool called Outlines, which enforces the structured generation and the output of a large language model. So the kind of workflow is the user asks a question in a natural language format. The large language model then takes the question from the user and actually converts it into the BioVelo query. So if you're familiar, kind of an analogy with SQL, It's kind of like having the large language model be able to generate a SQL query for you. And this is a more flexible approach than the RAG approach because it can make kind of arbitrary types of queries. And then from those queries, it will access the information from the database and give a response. Another thing I wanted to touch on here is kind of the idea of building benchmark data sets. So benchmarks test the performance of AI systems using a series of challenging questions and then grading the response. It helps evaluate the performance of large language models on domain-specific knowledge and tasks. And the reason I bring this up is because if you're someone who's coming from, a specific domain area expertise that's not AI or computer science, for example, the data in your domain is still very valuable for AI research and training. And so data sets and metadata that have been curated and labeled by domain experts is incredibly useful to help evaluate the performance of AI systems. So with Talk to Your Microbe, what we're working on is since we have we know the query language to generate queries of EcoPsych, we can programmatically generate hundreds or thousands of biochemical questions about E. coli and record the responses. In E. coli, all of the data is associated with evidence and literature, so it's been validated and it's a highly curated database, so we can trust the results in EcoPsych pretty well. The questions and answer pairs are then put into a JSON file, and then we can programmatically compare different types of large language models or even the same model using maybe some different tooling. As an example, we can compare like vanilla ChatGPT 4 with ChatGPT 4 plus the Talk to Your Microbe kind of tool, or ChatGPT plus the deep research mode as an example. And we can kind of compare the outputs of each of these different models and tools and kind of do a benchmark. So now I'm going to move away a little bit from E. coli and Talk to Your Microbe, which we were working on, and kind of explore a few other AI and agentic systems. The next one is called Reactome or Reactome. So Reactome is an open source, open access, curated and peer-reviewed pathway database, a reaction pathway database. Kind of in contrast to EcoPsych, which I talked about earlier, it emphasizes, human and related model organisms. So we have humans, rats, mice, pigs, cows, etc., as kind of the majority of where a lot of the information is. It has tools for pathway visualization, and you can interact with the pathways and also do different kinds of pathway analysis. And this is kind of led by a multi-institutional group. They recently developed something called React to Me, which is their AI chatbot. They use RAG to convert the data in this pathway database, Reactome, into several CSV files, which are then embedded into a vector database. They include several custom tools and agents as part of their chatbot. They have some tools to help check for the completeness of the answers, to check the language, to help reformat questions, to check safety, to search UniPro, the protein database, to do web searches, and then also to integrate the Reactome data. The main tech stack that they're using is LangChain, as well as ChromaDB for the vector database, and then Chainlit as the kind of front end. So here is just an example. I asked a question about this gene, TP53, its mutations, and kind of relation to uh, cancer progression. And so I asked this question, and it spits out a lot of information about the different pathways that are affected from this mutation of this gene and how that affects the development of cancer in a cell. And it also provides several sources uh, to kind of provide evidence for its responses. So I'm going to move on to another example. This one is called ChemCrow. And this is moving now from kind of biology and biological systems into more chemistry. So ChemCrow is an AI assistant for chemistry, and it's designed to help aid with chemical synthesis, drug discovery, and materials design. It uses a chain of thought reasoning loop using this REACT approach. So the idea behind this REACT approach is first the agent comes up with a plan and then does an action, which means it selects from a set of different tools it has access to. It will then use the uh, tools and then analyze the results of the tools. Um, it includes several different kinds of tools. There's kind of general tools such as a literature search, a web search, it can write Python code. Um, there's also kind of molecular type of tools to look at similarity between molecules, check for patents. And there's also safety tools. So to check the safety of the kind of chemical compounds being discussed to see if they're explosive or maybe on a list of unsafe um, chemicals or molecules. And the overall goal is to help with autonomous chemical synthesis and kind of further develop this human AI collaboration. And in this paper, they actually they explore actually using a fully autonomous kind of robotic experiment as well, kind of guided by this AI assistant. So as a quick demo here, I asked it to predict, based on one of their examples, the product of a mixture of two chemicals and explain the reaction mechanism. So it generates two of the kind of molecule schematics and then explains the mechanism. As you can see, this is a publicly available tool, and it has a front end that you can access on Hugging Face. And I asked it another question about synthesizing the molecule and how much it cost. And it kind of messed up here at the end of giving me the cost. I just kind of used X and Y instead of actual price values. So I wanted to show that because while this is a impressive tool, there's still always kind of room for improvement, and you should always check the output. And you can kind of learn more about this particular tool from these links here, which if you're interested, I can share in the Discord. And on Hugging Face, you can actually just demo it out. You don't have to install anything. So the final topic I'm going to close out on is a final agentic system that's being developed here at PNNL. And this is designed to be a teaching tool for domain subject matter experts to help demystify kind of black box approach of AI systems by using similar familiar scientific tools. And also to be for software engineers and data scientists to kind of illustrate how to connect different tools together in an orchestrated agentic system. So at a high level, the architecture involves a user accessing the Streamlit user interface, which connects to a LangChain agent, which is connected with a large language model. Excuse me. There's the LangChain agent is then connected to a model context protocol wrapper. I don't have time to go over all of the model context protocol. I think it's going to be discussed a little bit later today. But briefly, model context protocol is a way to standardize connecting different tools with different large language models. And you can kind of think of it as, similar to how we have a USB-C standard to connect different hardware to a computer. This MCP protocol allows us to connect different types of tooling to different types of agents in a kind of standardized way.
Excuse me, August, sorry to
Yep.
interrupt. Can you please wrap it up in 1 minute so we can have
Yep.
1 more minute for questions? Thank you.
Yep. So this is just an example of using the multi-agent workflow, and it's able to kind of produce a kind of a set of different tasks for this particular objective, which was to analyze a new drug target. So kind of to finish things up, I talked about a range of different AI systems, and kind of the goal of solving complex multi-step problems in biochemical research. And I think, this is an exciting area, and it's opening up some, interesting opportunities for human and AI kind of collaboration and scientific research. And with that, I want to acknowledge everyone on the team, Abby, Jeremy, Paul, and Yvette, and also the funding from this EMSL developer proposal. And thanks everyone for listening. And my contact information is here.
Thank you,
Thank you.
August. Great talk, great presentation. Do we have any questions here in the audience? We have time for a couple of questions.
Hi, I'm gonna just ask one part of my question. Like during embedding, the whole, all the files of your results gets embedded in your RAG documentation, right?
Yeah, so there are different approaches, but yeah, you will have a vector embedding where all your files are embedded into this database and indexed.